Another talk on offer is the physiochemical properties and what are they. So what will be covered in this talk? First, we'll be wanting to understand, is there any guidelines that drug discovery projects follow to monitor chemical progression? And when we talk about physiochemical properties, are there any important factors that we must consider? And one of these is actually permeability, and this is going to be a highlight of the following talk. But permeability isn't a physiochemical property. What is the physiochemical property? So one of them is lipophilicity, and we're going to understand, can this be controlled? And how do we incorporate it into drug design? Is there a give and take with other physiochemical properties? Highlighted, or at least related to permeability, is also pKa and solubility, and these two parameters will also be under study. We mentioned previous that we wanted to be able to understand, is there any guidelines? And some of these could be referred to as Lipinski's rules, which we will also be focusing on. However, to understand physiochemical properties, we must first start at the beginning. However, not really the beginning, more so the end. There is 95% attrition when it comes to clinical success, which is awful. There are so many projects entering the pipeline that just don't seem to make it into a clinical drug. AstraZeneca are aware of this failure and wanted to understand more about their own projects. And in doing so, they looked at 142 of their own drug discovery projects within a 5-year period from 2005 to 2010. And here are two graphs representing the data that they came up with. They discovered that upon entering into preclinical, a large number of projects were entering into preclinical and then quickly descending off when it came into phase 2 clinical trials. AstraZeneca took a look into their project failures for each of these individual stages. What you can see is a large number of these projects were closed due to safety. However, when it comes into phase 2 clinical trials, it appears that efficacy is actually taking over for safety. However, it must be noted that in order to comply with safety procedures, the dose becomes affected, and in doing so, the right dose to reach the efficacious effect could come into play as to why efficacy is a large factor when it comes into phase 2 clinical trials. What they deemed is that in 75% of these safety closures, it was always related to compound structure or compound properties. And what they wanted to do was they wanted to ensure that correct compound properties were coming into the pipeline to minimise the attrition later on. So it's all about safety. So where to begin when it comes to understanding compound properties? First of all, we need to know what kind of drug are we working with. And the following talk is going to be mainly focusing on oral drugs. That's what all drug companies are trying to aim for, is something oral. You might already be aware. Here's the human body. I hope you're aware. How does an oral drug make it into the body? It's first taken in via the mouth, and then it transports itself down to the stomach. Falling from the stomach, you get absorption through the intestines, which takes you into the hepatic portal vein and thus leading you to the liver. From the liver, if the drug survives the liver anyway, you then reach into the systemic circulation, where you are then able to cause your efficacious effect. What properties are on focus here? There's absorption, distribution, metabolism, excretion, and toxicity. And there is one compound physiochemical property that relates to all of these factors, or at least one that's going to be mentioned in this following talk. However, we're still sticking on the broader overview, the basics first. So, what can be controlled through compound design? We can create what is known as a covalent inhibitor. We know that when we take an enzyme and a drug and we put them together, that they form this complex that is perhaps not breakable, and it causes your desired effect in your disease model. Or you can get what's known as a competitive inhibitor, where your drug is able to release itself from the site or the enzyme target and then go back and forth between the two to cause its effect. However, you can also get a drug that causes a toxic effect, and this is something that we don't want. And how do we control for these toxic effects? What guides do we use in order to understand our compound design? So before taking a look at that, we must focus on the ADME properties. Oral drugs, what is its biggest factor? And that is permeability. And the permeability that we're going to speak about in the talk is actually passive diffusion. There are a number of different ways that drugs can pass through the membrane. This can be paracellular, it can be endocytosis, it can be active transport. But for the purposes of this talk, we're going to be talking about passive diffusion. And first we want to be able to look at the phospholipid bilayer and get an understanding of it. The phospholipid is comprised of a polar head group and a lipophilic tail. But looking into its chemical structure here, its head group in this is phosphatidylcholine. Here we have the choline head group. It's polar, it's charged. Therefore, we know that if it exists in this bilayer, the polar group is going to be facing into a polar environment to become stabilized. The head group is often attached onto a phosphate, and the phosphate is attached onto a glycerol backbone that is therefore subsequently attached on to its fatty acid chains. Moving on from here, you can have different head groups, and these are just a few of the more popular head groups. You can see here one exists charged and polar, whilst the other contains many, many polar atoms. What happens is that the polar head group is able to interact with its polar environment, often, in this case, water molecules. But how does something actually cross the membrane? So here you have your drug molecule, and it is being solvated through hydrogen bonds with your aqueous environment. The drug must shed this polar solvent, and it therefore then can cross over the membrane and then reforming into the other polar environment on the other side of the membrane. However, what parameters influence the crossing of this membrane? So the first being is size. Molecules of a high molecular weight cannot pass as readily through these tightly packed regions as small molecules can. We therefore then look at lipophilicity. Increasing lipophilic molecules means that they can become more polar through the nonpolar central core of the lipid bilayer, but too lipophilic and the drug can be stuck in these membranes. Too polar and the drug will not be able to permeate past. It should be noted that the highest energy barrier is where there are tightly packed, highly ordered region of the phospholipid side chains, which is near the glycerol backbone. That is the biggest energy barrier that your drug must overcome on both sides of the membrane, once entering and once leaving. The last of these parameters is solubility. Now here, solubility is actually depicted as a cartoon reference and done very simply. I do want to highlight that solubility is actually more complex than what is going to be outlined here, as this is not the focus of this talk today. A drug can dissolve into smaller drug molecules, and in doing so, these smaller drug molecules increase the surface area at the site of the membrane and can aid with the crossing over through the membrane, just as described when we talked about size, is that large molecular weight compounds can't pass readily through the tightly packed region of the membrane. Let's understand though lipophilicity. Molecular weight is kind of a given. Too big, not good. Small, good. So let's understand a little bit more about lipophilicity. Lipophilicity plays a massive role when it comes to understanding ADME properties and involved in almost every single one of them. To be lipophilic means that you're hydrophobic. You are consistent of often carbon-hydrogen atoms, and your major forms of interactions are van der Waals. So not ionic and not hydrogen bonding, it's van der Waals interactions. But what does something lipophilic actually look like? You can have something that's comprised only of carbon-hydrogen, something that contains a small amount of polar residues, something that contains a large amount of polar residues, or you can actually have a final drug molecule and there can actually be regions of polarity and regions of hydrophobicity. So to be lipophilic doesn't often refer to as the molecule as a whole. You can often refer to portions of the molecule where there are regions of lipophilicity incorporated in. So, lipophilicity. To have a high lipophilicity means that you are more hydrophobic in that region, or you are in general hydrophobic. This also means that you contain no or you have minimal ionizable centers. To be low lipophilic means that you are polar. You contain polar and or ionizable centers, and here is just a diagram which we will explain later on towards ionizability. What does it mean to be high in lipophilicity, low in lipophilicity? How can we actually measure if something is high and something is low? We have what is known as the partition coefficient, otherwise known as log P. And the two solvents that are under study are often octanol and water. You have a lipid environment and you have a polar aqueous environment. And here's the structure of octanol at the side. You can see here it has a polar region and a long lipophilic chain. It looks something similar to what we have discussed in previous, which is the phospholipid. However, not all drug molecules are neutral. Not all drug molecules have no or minimal ionizable centers. Quite a lot of drug molecules do contain ionizable centers. So how do we correct this log P value to account for these differences? It's what's known as the distribution coefficient, otherwise known as log D. And here your aqueous phase has been given a pH. It's been changed in some way, and often the pH at which it's measured at can be done in superscript next to where you find log D. It should be noted that it's not always 7.4, but in a large number of cases the log D is measured at 7.4. So how does this affect us measuring lipophilicity? What's too high? What's too low? What does it mean to be ionizable? How does this change my lipophilicity? How does this relate back to permeability? So as mentioned previous, almost all drugs have one or more ionizable center. Ions are stabilized in a polar environment. We know this. And we saw from the previous diagram that in a polar environment you can actually solvate with the polar environment, and you're therefore able to increase or enhance your solubility. But we saw from the previous diagram that if you are solvating with the outside environment, you're starting to become less permeable because you have to shed this interaction in order to be able to cross over the lipid bilayer. So the next important factor is not just lipophilicity, but it's also being able to understand what is pKa. And pKa is what determines your ionizability. Here is a simple diagram of an acid. Here we have the neutral form of the acid containing its proton, and we have the anion where it has shed its proton. At a pH, a neutral pH, here we have 7.4. We see here that there is a larger portion of the anion than there is of the neutral species. In doing so, it's only the neutral species that can cross this cell membrane. You can see here by the size of the acid and the size of the arrow that minimal amount of this is actually passing through the cell membrane. However, if you were to take an aqueous environment that is in favour of you forming this neutral species, you can see here that more of it is therefore able to cross the cell membrane, and in doing so you can now say that your drug is more permeable than what it began with at pH 7.4. But this doesn't really help us understanding what is pKa. What is pKa? pKa is the pH at which you have 50% of the neutral form and 50% of the ionized form. And here you can see by this diagram represented here. You have this formula, the neutral acid breaks down into its anions, and you can see here that as you increase the pH for an acid, you are actually minimizing the amount of the neutral species but favoring the increase of the anion. In doing so, your compound is becoming soluble, increases its pH because you're forming more of the anion, but you know that the anion can't cross the membrane, so you're actually making it less permeable. And the same is also true for the base. You can see it's just the opposite form of the acid. The base is in favour of a higher pH, the acid is in favour of a lower pH for the uncharged species. So between these two properties is about finding the right balance. What is the correct amount of polarity? What is the correct amount of lipophilicity? Does charge come into play? Here we have epinephrine. It has many polar centres as well as even a basic centre indicated here. Epinephrine was attempted to be used for the treatment of glaucoma, but they realised that it actually had poor corneal permeability. So you know, to aid with permeability, that you need to have something that's lipophilic. So this is what the drug company also did. They decided to seek out the centres that they could increase its lipophilicity. By sticking on these two ester groups, they managed to increase the lipophilicity, thus increasing the corneal permeability and marketing it for the treatment in glaucoma. However, can we look at another property? Solubility, remember. By increasing this lipophilicity, you could be influencing its solubility in the wrong direction. Remember, you need a balance of both. Here is a HIV protease inhibitor, and the only bit that we're going to look at is the R group here. You can see the R group starts to become less lipophilic as you move down. You can see this by the increasing number of polarity or potentially even ionizability through this pyridine nitrogen. You know that as you increase polarity or even the likely chances of ionizability, you are increasing solubility. However, the opposite is true for log P. In the opposite direction is where you see an increase in lipophilicity. However, which one is the right one? Which one should you be taking forward? Which property or physiochemical property, should you sacrifice? And the answer isn't entirely always true. It's mainly dependent on your target product profile or your target candidate profile. However, what I can tell you, the pyridyl R group is the one that led to the marketed HIV protease inhibitor indanavir. So we've understood about molecular weight and we have an understanding about lipophilicity. However, the last of which we want to have a look at is solubility. So solubility is actually required for intestinal absorption and oral bioavailability. We know this. We also know as well that solubility can be different in different environments. Why is this? Well, the differences between a buffer at pH 7.4 could be leading to ionizability. We don't know. It can be different from a biological assay media that contains 1% of DMSO. So you have to be very careful as to how you have measured your solubility. How do we influence solubility? So we saw in the previous diagram that through the addition of hydrogen bond donors and hydrogen bond acceptors, i.e. interacting with its aqueous environment, we can enhance its solubility. Here's a very simple case here. We have this molecule here that contains low solubility. By adding on this methylene hydroxyl, we can actually make it more soluble. However, that's a good case, and let's say that did make a drug in the end. However, when is doing this something bad? When is doing something good? So here we have a compound that is very, very soluble, possibly because of this diacid here. However, this compound suffers from low permeability. And why is this? Well, these acids could be charged. We know that ionized species can't cross the membrane, thus leading to its low permeability. However, by giving a little sacrifice of solubility, by adding on these ethyl esters on both, we actually are able to greatly increase its permeability because now this molecule can pass through the membrane as it's no longer or it's minimally charged. So what are the different properties that are influential for solubility? Well, we talked about lipophilicity. Too lipophilic and your molecule is not going to be soluble. However, too lipophilic and your compound will become stuck in membranes. The right lipophilicity and it will pass through. You have then size. You have pKa, the ability to form ionized species, and you also have what is known as a crystal lattice energy. Here is the solubility equation depicted by log S. Not going to go into too much detail about it in this talk. However, what you should be noted is that as you start to decrease solubility, you see a rise in two other properties. It's log P, its lipophilicity, and its melting point. Melting point in this case is referred to the crystal lattice energy. This means that as you start to favour this crystal lattice formation, you then start to reduce your solubility. So what do we know? Drugs have to be of a particular size to cross the lipid bilayer. But what size? We didn't discuss. Is there any value that we should be looking out for? We don't know. Drugs have to be in an unionized or neutral state. But how do we know what state they will be in? How do we know if they can form this ionized state or unionized state? We also need to exhibit reasonable solubility, but we've talked about how to achieve this, but we don't know what's too much or what's too little. And how can drugs be designed with this awareness? This is where Lipinski came into play. Lipinski took a study of oral bioavailable drugs, and in this study he undertook just over 2,000 compounds. And what he did is that he used phase 2 selection process as a filter. And why phase 2? Insoluble and poorly permeable compounds would have been eliminated from compounds that survived on entering into phase 2. So they know that compounds entering into phase 2 should have correct permeability and or absorption to be able to enter this phase. And how do they decipher this? By using the USAN or INN names that are given to oral drugs when they enter this phase is how they selected them. And what did they discover? They discovered a number of things, but they discovered 4 properties that kept on creeping up. These were molecular weight, these were lipophilicity, hydrogen bond donor, and acceptor. Now, do you see a pattern here? The size is influential to permeability. The lipophilicity is influential to permeability, and its ability to solvate or interact with this aqueous environment, aka solubility, is also a factor of permeability. And by undertaking this study, Lipinski found that drugs that fell within this criteria appeared to make it into phase 2 and believing to have good permeability and or absorption. This is known as the rule of 5. However, much conflict later on has deemed it, yes, it's still known as the rule of 5, but it's actually a guideline more than a rule, because this can be broken for some drugs. However, it is said that if 2 or more of these rules are broken, you are increasing the likely chance of your compound suffering from poor absorption and poor permeability. So using these rules, what do we start to understand about our drug matter? Well, we start to say, okay, as we start to move through the drug discovery pipeline, we see that we start to increase its molecular weight and increase its lipophilicity. But Lipinski has given us a set of guidelines, parameters that we must stay within. And you can see here, Lipinski's rules given here by molecular weight of 500 and a log P of 5. You can see the optimal drug space still falls below this parameter. We know that as we start to build upon our chemical matter that it gains in molecular weight as well as lipophilicity. However, what we must be aware of is that increasing these factors, in particular the lipophilicity, you can lead yourself to some toxicity issues later on in the process. So it's something just to be aware of. That, yes, the log P of greater than 5 is leading you to permeability issues, but subsequent studies later on have shown that a log P of greater than 3 could enhance your preclinical toxicity. So this is why Lipinski has now been known as a guideline rather than a rule. To enhance upon that effect of toxicity, the reason being is highly lipophilic molecules are able to concentrate themselves in plasma membranes, and we know that plasma membranes contain many signaling systems, and in doing so, the lipophilic molecules being sequestered here is thus potentially able to enhance toxicity. We've been given a guideline. We've been given Lipinski's rule of 5. That's fine. But are there any other ways to try and help us to determine what sort of physiochemical properties we need? Or how do we actually measure we're doing something meaningful and right? And this can actually be undertaken with some metrics, such as ligand efficiency. However, for the purposes of this talk, details into this have not been undertaken, but will be provided in subsequent talks. So to finish upon this point, what do we know? We started to undertake the journey of physiochemical properties in drug design. What are they? So one of the major factors we started to follow was permeability and absorption, and why? We are interested in oral bioavailable drugs, that's why. And what properties mainly affect these? We found it was size, we found it was lipophilicity, and we also found out it was solubility. What did we start to study? Well, we only focused in on passive diffusion here, and we started to understand these different physiochemical properties and how they affect this passive diffusion. We spent a lot longer on lipophilicity because it has a greater influence on its ADME properties than most other physiochemical properties. We discovered that too lipophilic a compound can result in sequestering in the membranes and thus making the compound not able to come out onto the other side of its membrane. We found as well that too polar, that the compound just won't be able to permeate. However, polarity is needed for solubility, so there must be a balance of these two properties. We discovered that if you are to reduce this hydrogen bond donor and acceptor, that you are able to aid with permeability, but again, that leads into solubility. These two factors are required to get your drug to be soluble. Large molecules find it hard to cross by passive diffusion. And the reason being is because they just can't seem to pass through the tightly packed region of the lipid bilayer, as a small molecule would. Lipinski gave guidelines, known as the Rule of 5, to aid you with its physiochemical properties: molecular weight, lipophilicity, hydrogen bond donor, hydrogen bond acceptor. Lipinski gave guidelines, the Rule of 5. However, why are these guidelines? Subsequent studies later on have shown that they're not disproven, but there are other additional factors that we have to take into consideration, such as toxicity. We discovered that too lipophilic a molecule, like a molecule with a C log P of perhaps greater than 3, could actually maybe enhance some preclinical toxicity later on. It's important to know that physiochemical properties are important for drug design and hoping to try and minimise attrition later on in the discovery process. Safety is a massive factor when it comes to creating a new drug for human intervention. This talk was brought to you by the Wellcome Centre of Anti-Infective Research, the University of Dundee, Scotland, UK.